Good afternoon. It's now my pleasure to introduce Dr. Devin Smith, CEO of Arbor Bio and Chairman of ARM. Devin, thanks so much for joining us today. And to start off, can you maybe tell us a little bit about Arbor Bio for those new to the story?
Yeah, happy to, Gula. Thanks for having me here, and always look forward to these kinds of discussions. So Arbor is a next generation editing company. We were co-founded by Fong Zhang and David Wall, Fong being the CRISPR pioneer and David an expert in high throughput automation who co-founded companies like Illumina. And they got together, and Arbor was founded on an idea rather than IP. And that idea was Cas9 was the first nuclease found, but not necessarily the best. And why don't we go into the metagenome and identify novel, differentiated, better characterized editing technology? So with that, we built a very elaborate discovery platform that utilizes a mix of machine learning and AI-driven approaches for search, discovery, as well as engineering novel editing technologies. We feel we have the most extensive toolbox of proprietary editors in the industry, which allows us to really focus on bespoke therapeutics, where we can really understand the underlying disease pathology and then bring to bear the right tools to bear. We are focused in 4 areas in editing that really allow us to cover all the known genetic diseases today. In addition, we've got a very experienced team that I think it's important that you have a talent pool that understands how to bring novel therapies like this into the clinic. And we're really focused ourselves on in vivo editing. We partner on the ex vivo side, but in vivo is where we want to focus. And that's allowed us to focus in on the liver initially, where we can deliver via LNPs, and then shift to CNS, where there is tremendous unmet need and the opportunity is immense. The goal is to have our first IND CTA in the next 9 to 12 months, which would be the first non-Cas9 nuclease tested in vivo in humans. And we've raised over $300 million today. We've got some important in vivo partnerships with Vertex, as well as some ex vivo partnerships with Vertex, Edagene, and others. The only other note I would make as we think about where we focus is, if you think about the broad world of genetic diseases, there's kind of 4 things you need to do to correct or fix or treat a genetic disease. You can stop or knock down a protein that you've got too much of. You can rewrite portions, SNPs up to say 20 to 40 base pairs where you may have hotspots and mutations to correct mutations. You can excise areas where you may have too much of something, like a triplet repeat, or you can knock in an entire exon or gene into an endogenous locus. So those 4 areas are where we really focused our technology, and that's all predicated on having this set of chassis, these nucleases that allow you to sort of plug and play different effectors on them to enable you to do these 4 things. So that's where we're focusing. We're not doing base editing. We're also not doing safe harbor large insertion. So with that overview, Gula, I'll turn it back to you for questions.
Perfect. Thanks, Devin. So in terms of manufacturing, can you discuss the type of therapeutic modalities that you'll need to make to advance your pipeline?
Yeah, so there's, with our, if I break it into 3 buckets, so there's the ex vivo side. So because we're not developing our own ex vivo cell therapies, that means that our partners do that. And so we provide our technology, they manufacture, so we don't have to do it. That is on purpose. I think it is difficult for a company to manufacture cell therapies and genetic medicines. I think it's there's very little, if any, synergies on the CMC and manufacturing side. So we'll set that bucket aside and focus in on the in vivo medicines. There we've got 2 types we have to manufacture. For liver, we have to manufacture an LNP containing a messenger RNA, which encodes for the nuclease technology or editing technology we're delivering, and a guide RNA, which tells the nuclease where to go and what to do. And so for manufacturing them, we need to synthesize a guide. That's done, our guides are between 20 and 50 base pairs, so quite a bit shorter than, say, a Cas9 guide, which can be well over 100. We then have to make a in vitro transcribe, an mRNA, which length varies depending on the length of the nuclease. Ours are very small nucleases, about half or less the size of Cas9. So it's smaller mRNA you need to in vitro transcribe. And if you add an effector, it may make it longer. So those two components are then packaged into the LNP, and then that is your drug product. On the CNS programs, our initial programs are actually going to utilize an AAV. Their manufacturing, obviously, you've got to manufacture the AAV. Inside that AAV will be the DNA containing the nuclease, as well as the guide and any other pieces you need to place in there. Because again, we have very small DNA effectors, we can place a nuclease plus, say, a reverse transcriptase into a single AAV if necessary to do that. So those are the two sort of manufacturing modalities we have, with the LNP being near term and AAV being the sort of in the midterm.
So in terms of manufacturing challenges, are there, we've talked a lot about AAV at this conference, talked about editing in other contexts as well. What are the main challenges that you see associated with manufacturing each of those components?
Yeah, I think the first decision to be made is, do you do it inside or out? And while inside makes sense for perhaps animal experiments or early quick discovery work, all of our work for studies that really count is done outside. So with that lens, on the challenge side, I think it's making sure you pick a vendor or set of vendors that have experience with LNPs, experience making mRNAs, as well as guides. And you will not find someone that can do all three. And so it's finding the right partner that can allow you to at least minimize that supply chain. That's always a challenge. Secondly, I think being careful that if your mRNA or guide get too long, that can be a challenge to synthesize. And so making sure that, with our technology, we have relatively modest guide length. If you get over 100, it can sometimes be a little more challenging to synthesize the guides. Or if your mRNA gets too massive, that can also be a challenge. So those are some of the challenges there. I think the other piece is making sure you have consistency in your LNP formulation. So the consistent, similar to what you do with AAV, a consistent batch, so that the analytics are within range that you desire when you manufacture. AAV, I think the challenges, you probably talked about all of those, are pretty standard, I think, across our space. Making sure you don't have large numbers of empties, making sure that nucleases are unique and that you have something that likes to cut DNA. So during the manufacturing process, making sure that you don't inadvertently chew up your DNA because you have a nuclease present. So these are some of the things that, I think from a manufacturing perspective have to be dealt with. I think that the other element is identifying capsids that, some of them are extremely well validated from a manufacturing perspective, like AAV9, while the more novel capsids may have a more challenging manufacturing route. So that's at a high level, some of the challenges we've seen.
Mm-hmm. Yep. So in general, genome editing has been able to incorporate a lot of the learning from other genetic medicines modalities. So how can you apply some of the manufacturing learnings? Obviously, there's been a lot that has been done on AAV. Obviously, lipid nanoparticle and mRNA delivery has, scalability has been tested extensively. So to what extent can you port a lot of these learnings, and how much process development needs to be done for kind of individual programs there?
Yeah, it's interesting. If you'd asked me that question 5 years ago, it'd be a very different answer. But I think today, obviously, from an LNP perspective, the COVID work has greatly enabled us and anyone else using LNPs to where there is capacity. There's a tremendous amount of experience in thinking about how to make and formulate these things. So that's very beneficial for us. And so we can learn a lot from prior work. On the AAV side, again, I think it's similar, where because we've got approved therapies, a tremendous number of therapies in the clinic, for us, it's leveraging that expertise and experience to make sure that we don't repeat the mistakes of the past and really learn from that. And that I think is an important thing for us as a company throughout, is making sure we learn from others so that we don't spend time and money redoing things that have already been learned and they know they didn't work. So that's been an important, thing for us to make sure we continue to try and do.
And you kind of touched on this earlier, but anything else on this front in terms of the ways in which having a genome editor as a cargo changes anything regarding manufacturing considerations?
I think in LMP it does not. I think in an AAV it can, and so again, depending on what you're delivering, it can cause challenges in manufacturing because it may inadvertently start chewing the DNA of the cells or of the packaging cell line, or you may, so you may impact your yield of your AAV output. In addition, I think that's one potential challenge. The other is if you are delivering something that likes to cut DNA, which is fantastic because that's its role. But how do we make sure that we, once it's in the cell, though, it's not there permanently? And so there's some other technology and tricks to make sure that, because it likes to chew up DNA, that you can work on self-inactivating approaches as well. So AAV is a little more complicated, but I think there are various approaches to making sure you can adequately manufacture an AAV containing some sort of gene editing modality.
Makes sense. And so again, you touched on this a little bit regarding the decisions with respect to internal versus external CMC and manufacturing. Can you expand a little bit on the decision process there, what you plan to do internally, and what do you plan to do with CDMO partners now versus, let's say, a few years down the line when you're further along in clinical development?
Yeah, it's always an interesting challenge, I think, for companies to decide where do you do inside versus outside. I will say, I think, I've never been a fan in early stages of making it inside for talks or clinical in early stages, because it's a very capital intensive just to build the manufacturing, not to mention the ongoing OpEx. So that has not been something we have focused on doing. I think making sure you have the right PD and analytical expertise in-house is critical. So we make sure we have those expertise in-house. We know how to do that, process development. We know how to do the analytical assessments of whatever it is we're manufacturing. That's important. But as far as the actual nuts and bolts of GMP clean rooms and such, that's all outside. And for us, that has been a strategic imperative. I think at some point, there are elements we may want to bring inside. I don't know when and what that might be. It really depends on how things evolve. I mean, currently there's a fair amount of excess LNP capacity. I think there was a lot built for COVID, and that's all sort of opening up. There's also a fair amount of excess capacity in the AAV space because I think it was overbuilt. So today that we don't feel a need to do that. At some point when we're peri-commercial, we may want to do that to own our capability to deal with cost of goods and impact that. But I could see that probably on the AAV side. I don't know if on the LNP side we would or not. It really comes down to sort of what's the overall cost and efficiency profile look like externally versus what we could do inside. But that's a future problem to deal with, and I think minimizing the capital and OpEx internally we spend on this near term makes a lot of sense, and I think is something I think I've been consistent in for the last decade. I just think it's challenging to build your own in-house manufacturing when you're an early stage clinical company.
And so you touched on this a little bit earlier, but what do you look for in a CDMO partner? I think you mentioned the supply chain.
Yeah, I think
there.
It's an interesting question. As you work with CDMOs over time, you recognize that each one has things they're good at and things they're less good at, and none of them have the full package. And so it's really knowing what they're really good at that is a strength that benefits your program, and then also knowing what they are not very good at so you can supplement that as needed. So that's an important piece of it for us. And so as we go out to make decisions on CDMO partners, we have to be very thoughtful about that piece. The areas that really make a difference from our perspective, one is who's the project manager on that side? Who's the person who's on day-to-day point that will be the advocate for your company within their organization? I think finding a CDMO that's willing and to have a person in plant during manufacturing is also very important, and also during the tech transfer process, so we can help teach them and make sure they're doing things appropriately. And then I think also finding a CDMO that has internal analytics expertise, as opposed to shipping everything around the world to have your analytics done, and it takes 3 months to get your analytics back. That, I think there's pieces of that help you minimize the time frame for GMP manufacturing, also the time for getting your product released. And then finally, I think finding a CDMO that has experience in manufacturing for clinical studies, both in the US, Europe, and or Asia. So they have comfort, they've done it before. So those are the pieces we look for. And there's, I think today there's a surprising number that are pretty good. None of them are perfect, but there's a surprising number compared to, say, 5 or 10 years ago.
Interesting. So on that note, kind of in terms of the trends that you're seeing, perhaps from your perspective as ARM chair, what are you seeing in terms of trends regarding manufacturing, either with regard to capacity or other elements?
Yeah, so I can speak. So from an ARM perspective, I think ARM has really worked hard to try and identify areas where all the 500 member companies in cell and gene therapy can work together to solve problems, the common problems. So there's been a fair amount around manufacturing processes, making sure we try and standardize approaches where it makes sense. Some of these things are critical. I mean, where we're seeing trend-wise, I think you saw a trend over the last, call it 5 years, where everyone wanted to make their own AAV in-house and build their own little GMP suite. That trend has done a 180. I think a lot of those are now up for sale and being sold off. I think other trends that we've certainly seen, I think, is the ex vivo space as well. So cell therapy generally, I think you've seen a real shift there on autologous versus allogeneic and where it's manufactured. I think that a fair number of folks were trying to do it in-house. I think that trend is also reversed somewhat, as you've seen, just some of the capital constraints that are showing up. So, those are some of the trends. I think it's the companies that are successful, are able to navigate through whatever whatever the issue is, whether it's they've made a decision to in-house and they figure out how to live with it, or they find a partner to take over it, as some companies have done. So I think it is a very dynamic time right now, as in the cell and gene therapy and editing world. But I think you're starting to see some companies whose strategies are paying off because they've been very thoughtful in how they allocate capital.
Got it. So you mentioned some of the changes in capacity and capacity constraints in different segments. What's the case for the ex vivo space? Is there also kind of a greater build out in terms of capacity there?
Yeah, I think there's been quite a build out. I think you've got two pieces on both AAV and on ex vivo cell therapy, where you've had a massive build out on the CDMO side, as well as on the company side of GMP capacity, a lot of which now on the company side is being sold off to CDMOs or to new startups because folks are trying to preserve capital. I think that's in both the ex vivo and the AAV space. I think on the CDMO side, I think we've seen, I think AAV manufacturing tends to be very capsid dependent. I think we've learned over time that companies that can make AV9 really well may not necessarily be the ones, they may not be able to make AV5 or some next gen capsid well until they've have experience doing it. on the ex vivo side, I think, certainly, my prior company was in that space. I think you've seen a lot of newer startups come in with smaller suites. They say they have much, more focused expertise versus sort of the larger incumbents. I don't know if that is accurate or not, but there seems to be a lot of startups. You go to these meetings, like the ARM meeting on the Mesa, and there's a tremendous number of small CDMOs that I'd never heard of, that are now pitching their wares. So it's a dynamic space. I think there is capacity. 2 years ago, the capacity was quite constrained. I think today, between the mix of companies struggling and either terminating clinical studies and extra build, I think there is a little bit of extra capacity out there right now, which isn't a bad thing.
Mm-hmm. Got it. And so we touched on kind of challenges relating to manufacturing for gene editing programs. What do you see as kind of more broadly the biggest challenges in cell and gene therapy manufacturing these days?
I think the analytics side is always a challenge, particularly when you're manufacturing any sort of, living cell or quasi-living viral particle. How do you make sure you've got consistency, and how do you measure that? I think is always a challenge. How do you measure consistent potency of these therapeutics? I think is a consistent challenge. And how do the regulatory agencies view that? Because they may view 20 different cell therapies that are all T-cell related, and each one has a slightly different potency assay. And how do the regulatory agencies view that? What are their concerns? So I think the regulatory piece is not trivial, and it continues, I think, to be something we're all learning as we go, whether it's in cell therapy or gene editing for that matter.
And looking ahead, how do you think the space will evolve in the next 5 years?
So I'll start with editing, where Arbor is. I think in editing, I think we're going to see many more clinical trials underway, and those will also be underway in the US. I think we'll begin to see some real a tremendous amount of clinical data being read out across many different therapeutic areas and modalities in editing, I think, which will be very exciting, because I think editing is a modality that I think is here to stay and will impact human health significantly over the coming years and decades and centuries, perhaps. On the ex vivo cell therapy side, I think we're starting to see approvals of successful therapies. I think the Legend therapies has been doing extremely well, and it's really exciting to see we're truly impacting a lot of patients now in the oncology space with these, with the many approved therapies. In gene therapy, this is going to be a big year. I think there's multiple, I think 5-ish or so gene therapies slated for potential approval. So I think, again, another big year for, I think, the space that hopefully will continue to build investor confidence, as well as change the lives of many patients.
Great. I guess just one last question in the last couple of minutes regarding the FDA. There's been a recent transition where the FDA Office of Tissues and Advanced Therapies has transitioned to the Office of Therapeutic Products, and there's been overall some changes in organizationally in the FDA. How do you expect this, along with kind of various other comments from regulatory officials regarding moving forward manufacturing and otherwise streamlining therapies for rare diseases, to impact program development in the space?
Yeah, I think it is something that is, I think once the FDA, fixes the current sort of people crunch there, I think that, need to hire people. The PDUFA will help them. The recent PDUFA will help them do that. I think there is appetite to think about how do we become much more efficient in these novel, these rare and ultra rare diseases. So, for example, from an editing perspective, if we were to target liver or multiple of these ultra rare diseases, theoretically, all you would need to change is your guide. And if that's all you're changing, everything else is the same, how do you build a IND package that's sort of I&D light, so that you're not having to spend millions and millions of dollars for each new one where it doesn't justify, the ROI isn't justified. So I think there's a lot of interest in thinking through that. So because these treatments, as we get better and better at the larger, quote unquote, larger rare populations, we can move into these rare and ultra rare, but only with help from regulators to help think through how do we do this safely, but also efficiently.
Great. I see we're at the end of the session. So thanks again, Devin, for your participation, for the great discussion.
Thank you, Gil. A pleasure to be here, and thanks, everyone.